Breaking the Wall to Community-Led Climate Adaptation. Roberto Torres, La Ciencia al Toimón, Spain.
Hello. Imagine, what if I tell you that a significant amount of the knowledge about how organisms adapt to the environment and how they have adapted in the last 20,000 years fits in this little tube. You can have one in there, in your table, the Jody. But moreover, in a tiny little bag, you can see it from over there, but easier to see here. In that little fly is a Drosophila melanogaster, a fruit fly, that is an excellent model organism to understand climate adaptation, because like humans, their origin is in Africa, and they have moved all over the world and adapted to all the environments in only 20,000 years, a very short-term evolutionary speaking. So our challenge is to engage young people in the schools, and farmers, and other stakeholders in rural areas to commit and help us to identify adaptive mutations, the molecular organisms, the phenotypic traits, and the environmental variables that drive adaptive mutations, to understand how humans, animals, and plants can adapt to so different and diverse environments, like pressure and high altitude, and extreme climates. Sound familiar nowadays, right? So it's a very valuable knowledge. But we need to engage them in basic research, in molecular biology, long-term results. So it's a big challenge for us. So with Josefa González, the evolutionary and functional genomics in my organization, we designed this Melanogaster catch the fly, a citizen science network on adaptation genomics. First of all, we develop educational materials and resources to outreach activities and workshops to raise awareness in students and teachers, not only about the relevance of these scientific projects or the scientific approach, but mostly the relevance and the importance and the value of their contributions. So they can collect and classify biological samples properly for us. So that was in 2016. The first year, we started in Spain. So they collect and classify the sample for us. They send it to Spain. We sequence the samples. We got tons of big data and distributed to the European Drosophila Consortium on adaptation genomics. 72 labs in 29 countries related to this project. So it was a great opportunity to bring cutting-edge science to rural areas and implicate them to expedite a very important scientific process. So that was 2016. Since the second year and nowadays, we have built a very committed and proactive community across Europe, now in America, and there's more countries coming. They not only collect and sample now and classify now, they also get involved in all the scientific processes, stages of the project. Experimental validation, scientific community, technology development to create tools, bioinformatics, outreach, dissemination of results, and policy making. What we learned: do not patronize your audience. Push them to the limits. They are capable to anything, and peer-to-peer communication is powerful because we empower them with evidence. They create their own new narrative that they are not tellers. They are ambassadors and creators of new evidence and arguments to fight for climate adaptation. And they also help them to contribute to understand how climate change are impacting their local diversity. So for me, this is now. So you're gonna join us. Later, you can join our project. We have some starter kits. You can ask for them. And you're welcome to join us. Thank you.
Hello.
Do we have any questions? Yes.
Oh, thank you so much. So just to clarify, people are able to identify mutations that have phenotypic traits, and how many have you found? Have you found, what's the science beneath this? Have you found many mutations that you can relate to environmental changes?
Yeah, this is the research of Josefa González at the beginning. They work with transposable elements. So this is pieces of DNA that jumps and create new mutations, and they identify the ones who help the organism to adapt. So they collect, and with the sample they collect, the scientific community process the data. They turn back the data, and now they're starting to validate experimentally one part of the process, and also they are working now introducing themselves in bioinformatics.
So yeah, okay. I'm sorry. I'm always taking Jerry first. So yeah, we have here. Okay.
Just a quick question. Apart from being here at Falling Walls, how do you manage to reach out to new participants all over Europe and America?
Pardon me? Yeah, all the world. We're looking participants all over the world.
How do you manage?
We started with a scientific community, but everyone has a friend or a relative that works in a school, or in the hometown, in a village, at the beginning that way. And now people is approaching to us. We make some calls time to time. We don't make these massive calls because we're going to be able to attend everyone within quality, nor quantity.
I think we have a question. Yeah.
So once you find some mutation in some of these genes from all over Europe and so on, how do you validate them? As in, how do you relate them to environmental factors?
Okay, there's a big scientific community. There's different approaches. So there are, for example, there are 72 labs. They use all this data, and they have different approaches with the same organisms. So there are publications. They're contributing in the sampling and the classification. Classification is part of the validation of the students, but it's an ongoing project, very powerful. So in the part of the students, they contribute in the schools. We have designed a mobile molecular laboratory, and we give them some samples, and they have the mutation, and they validate a part of the project because they want to keep involved. Because the people who cannot go to the field want to get involved in the city in some way.
Yeah, we have only 10 seconds, so I don't think. Yeah. Yeah, no, 5 seconds. Sorry, it's so hard to say no, but yeah.
There's Josefa González, the scientific director. You can ask a lot of questions. Thank you.
Thank you.